Dear students, let us continue our study in this paper, which is Forensic Chemistry and Explosives, and in this particular module we shall study about how the examination of some common products can be done. And what are these common products? They can be edible oils, the gold ornaments, petroleum products, and the examination of alcohol in liquors that we will be focusing in this module. So, if we try to look at the learning outcomes of this module, after studying this module you shall be able to know about the significance of some common items of daily use which we use so commonly, and then how their forensic importance is there, and as a forensic scientist you will carry out the examination. And yes, obviously then after that you will be able to know some procedure of the laboratory examinations that you have to conduct. There was a time when food and daily use items were only used for their genuine purpose, food materials to meet the food necessity and the other items for their respective usage. But as the human civilization has developed, so has the criminal mindedness. In order to gain more monetary benefits, illegal activities have originated out from the vivid minds. They started adultering in the various daily use items with extraneous substances to fulfill their felonious intents. However, the adulterations were so naive that they were reluctantly tolerated by the society, as those defilements were fairly noticeable and be removed by handpicking. But as its criminal psychology which motivated to generate other innovational activities which prevented a layman to identify the adulterants. Still, some of the items were not decipherable from the beginning, such as jewelries, petrol products, dairy products, etc. Jewels and ornaments have fascinated the woman from the almost advent of the human civilization, and which makes them more vulnerable for criminal activities. Thus, the manufacturer may add other metals of similar texture and color in the gold or silver items in the process of manufacture. Similarly, due to the international crisis of petrol products, due to its scarcity and limited resource and obviously higher cost, they are frequently mixed with various other chemical compounds. In the case of alcohols, they are easily available in the market to every man. What makes them so open to misuse? The answer is the laws and rules of the respective states. Liquors are manufactured only with prior permission from the governing authority and the state administration. In order to escape from rules and regulations and procedures, one may clandestinely manufacture liquor by the limited resources which have been deprived to him due to its ineligibility. To overcome the shortage of resources added with the aim to earn more, cheap substituents are being added as adulteration. And this problem is not only limited to the illegal manufacture of liquor, but also it is frequently observed in the country made liquors. This may not be intentional, but the contamination is though a menace, and one may do it is unknowingly, but that can be deadly. Like if there is a methanol added, then it is highly poisonous and can lead to blindness and even death. So, now coming to the forensic issues of this adulteration. Oils and fats examination covers a major part in forensic chemistry, as these are often adulterated in many ways, which are adulteration of lower grade, diverse origin, or misbranding or contra-branding of products. The cases as above may occur recurrently, as oils and fats are extremely used as cooking means, and also in various industries like paints, varnishes and pharmaceutical industries. There may be theft cases or unlawful possession associated to oils and fats. In such cases, the samples are frequently referred for their examination for different purposes under the Essential Commodities Act 1955. The samples are very often required to be tested for various factors based on their physical and chemical properties of the individual oil and fat to determine the nature, quality, adulteration, etc. Similarly, in the case of gold, as it is precious and valuable metal, these ornaments are required their preservation, which includes repairing and polishing, etc. These exhibits are to be examined in the laboratory for analysis of gold. Equally, petroleum products, most frequently referred to any forensic science laboratory for examination, are petrol, kerosene, diesel, lubricating oil, furnace oil, waste oil, etc. And these samples are forwarded to forensic science laboratory for identifying their purity, detection of adulteration, if any. Liquor is normally known as a mixture of water and alcohol. Samples are seized by police and submitted to the forensic science laboratory for their examination, and the liquor is examined in the laboratory for two purposes. Firstly, for the excise purpose, where mainly the presence of alcohol plays an important role, and accordingly the examination of liquor samples for the qualitative and quantitative analysis is the main purpose of the investigation. And secondly, the liquor is examined for quality control or duplicate samples which are being peddled in the market. Let us now try to be familiar about how the examination of these is done, and let us begin with the examination of edible oil. And first of all, we will consider the detection of argemone in edible oils. And to find out this, we subjected to ferric chloride test. And how this test is conducted, let us try to understand. About 5 ml of filtered oil is taken and it is dissolved in 5 ml of toluene in a stoppered glass test tube. 5 ml of conch HCl is then added to it and it is then shaken vigorously and allowed the acid layer to separate. The acid layer is then separated with the help of a separating funnel to another test tube. 1 ml of ferric chloride solution is then added to this test tube through the side wall. The reagent and acid layer is then mixed well in the test tube and ferric in test tube and heated in water bath for about 10 minutes. On cooling, formation of straight or curved needle shaped reddish brown crystals in cluster indicate the positive test for the presence of argemone oil. The method is not sensitive enough to detect argemone oil if it is less than 0.1% level. Coming to now the detection of mineral oils in the edible oils, and this is done by taking about 22 ml of the 0.5 normal alcoholic QH solution. It is taken in a conical flask and 1 ml of the suspected oil sample is added. It is then boiled till the mixture becomes clear and free from any oil drops on the side wall of the flask. The whole content is then transferred into a wide mouth warm test tube. After that, 25 ml of boiling water is added along the side of the wall and it is shaken lightly. The appearance of turbidity will indicate the positive test for the presence of mineral oil. Next is the detection of hydrocyanic acid in edible oils. 15 to 30 ml of the suspected oil sample is taken in a 250 ml conical flask and about 50 ml of water is added and mixed well. To this 15 ml of 10% tartaric acid solution is added and mixed again. A stopper is put on the flask with a velvet cork from which a picric acid soaked paper is hanged out of about 75 mm long, wetted with a drop of 5% sodium carbonate solution. The flask is kept on a hot water bath by the side of the steam vent but not directly on the steam for 30 to 45 minutes. The red color on picric paper indicates the presence of hydrocyanic acid. Let us now move to the detection of cortal oil and soluble colors in edible oils. This is done About 5 ml of suspected sample is taken in some test tube and 15 ml of petroleum ether is added to each of these, followed by 5 ml of HCl of different concentration, that is 4:1, 3:1, 2:1 and 1:1 of HCl and water. Change in color indicates the presence of coal tar oil soluble colors in the sample. Now coming to the examination of gold ornaments after the examination of edible oils. In this we will be first analyzing the gold polishing powder. Generally the gold polishing powder is reddish in color and shows the presence of inorganic radicals such as copper, sodium, potassium, ammonium, iron, chloride, sulfate, nitrate, etc. The analysis of gold polishing powder is carried out by the procedure used for analysis of inorganic qualitative analysis. The gold polishing powder may also be tested for the presence of gold. Test for copper, this is a test with ammonium hydroxide. The test sample solution, which is the sample which is dissolved in water or HCl or nitric acid or aqua regia to form original solution, that is taken, and then ammonium hydroxide is added to it. If there is an appearance of blue color, it will indicate the positive test for the presence of copper. Then the next is the test with the Alizarin blue. A drop of reagent, that is saturated solution of Alizarin blue made in pyridine, is taken in a spot plate and to it a drop of test solution is added. A parallel blank test should also be run with water. Blue color appears in both the cases. Now 1 to 2 drops of acetic anhydride will be added. The color in the blank test will convert into yellow, but remaining of blue violet color precipitate in the test sample, if it is there, then it will indicate the positive test for the presence of copper. Then there is also test with glacial acetic acid. 1 to 2 drops test solution are taken in a depression spot plate and evaporated to dryness. A drop of alizarin blue reagent is added, followed by a drop of glacial acetic acid. Appearance of blue violet color indicates the positive test for the presence of copper. We can also conduct the flame test. Copper will give a bluish green color when it is tested by flame. Coming to the test for iron, first we have the test with potassium ferrocyanide. Few drops of test solution or the original solution are taken in test tube and to it a drop of conc nitric acid is added and then boiled for a few minutes. Potassium ferrocyanide solution is then added to it. If there is appearance of blue color, it will indicate the positive test for iron. Another test for iron is the test with thiocyanate. Few drops of test solution or original solution are taken and to it a drop of conc nitric acid is added and then boiled for few minutes. After that, ammonium or potassium thiocyanate solution is added. If there is an appearance of red color, it will indicate positive test for the presence of iron. After this, let us try to understand what are the tests for gold. First is the test with benzidine. How the reagent is prepared? 0.05% solution of benzidine is mixed in 10% acetic acid and few drops of the exhibit solution are then placed on a filter paper and added 1 to 2 drops of this benzidine reagent solution on it. If there is a blue color which appears on the filter paper, it will indicate the presence of gold. Test with Rhodamine B. How the reagent is prepared? 0.2% aqueous solution of Rhodamine B is prepared and that is what is the reagent. And what we have to do is few drops of the exhibit solution is placed in a micro test tube and you add 1 to 2 drops of HCl as well as 1 to 2 drops of Rhodamine B reagent solution and then mix them properly. The test tube is further shaken with about adding 8 to 10 drops of benzene. Appearance of red to violet to pink color in the benzene layer will indicate the presence of gold. After about 1 to 2 minutes, it displays an orange fluorescence if it is observed under the quartz lamp. Then there is another test which is the test with oxalic acid for gold. An appropriate portion of the exhibit is generally heated till NO2 fumes are removed. This solution is then made alkaline by adding sodium hydroxide solution. Solid oxalic acid is then added and heated. Brownish black precipitate, if it is formed, will indicate the presence of gold. Now, after gold, let us look at how the examination of petroleum products is done. And for the examination of petroleum products, you must have seen in the petrol pumps also, they say there is a filter paper test. Let us try to know what is this filter paper test. So, this is the filter paper test for the detection of kerosene or diesel in petrol. Petrol, you know, is highly volatile liquid fuel. It is a clean fuel and hence drops of petrol vaporize extremely fast and they vanish without leaving any trace behind. Kerosene and other high boiling compounds are comparatively less volatile as compared to petrol and they will not vaporize quickly. Hence, if petrol which contains kerosene or diesel as adulterant is placed on any filter paper, petrol will evaporate leaving behind the kerosene or diesel, which looks like oily patch with a characteristic smell of kerosene or diesel on that filter paper. If petrol is adulterated with volatile hydrocarbon solvents like aliphatics, aromatics, naphtha, then this test may not be detected by the adulteration. So, this you have to keep in mind. Now, coming to the examination of alcohol in liquor, there is a test for ethyl alcohol, which is iodoform test. Appropriate amount of sample, which can be distilled or as such depending upon the nature of the sample and concentration of ethanol present in it, is taken and about 1 ml of 5% sodium hydroxide solution is added to it. And then you add iodine solution by mixing 20 grams of potassium iodide in 10 grams iodine in 100 ml water, and you keep on adding it dropwise with shaking until the liquid persists the reddish brown color. It is then kept for 2 to 3 minutes. If the iodine color disappears, more drops of iodine solution is added until there is a persistent brown color of iodine. Few drops of dilute sodium hydroxide solutions are then added to remove the extra iodine. Equal volume of water is added and left for 10 minutes. If there is formation of yellow crystalline precipitate, it will indicate the positive test for the presence of ethanol, and this is based on the iodoform reaction or the iodoform test. Then there is another test for ethanol testing, which is the dichromate test. About 1 ml of appropriate amount of the sample is added with about 0.2 ml of 2% potassium dichromate solution, followed by 1 ml of concentrated sulfuric acid. The yellow color of dichromate, if it changes to green or blue color, it is because of the oxidation of alcohol taking place. This is the oxidizing agent. So, if there is formation of green or blue color, then it will indicate the presence of ethanol. Then let us see what are the tests for methanol. For methanol, there is a chromotropic acid test, and how it is done. About 1 ml of appropriate amount of sample is taken in a test tube and about 2 ml of KMnO4 solution, which is made by mixing 3 grams KMnO4 and 15 ml of phosphoric or orthophosphoric acid in 100 ml distilled water. This is taken and then shaken well. After this, few crystals of sodium bisulfate are added with shaking till disappearance of the color, and then 1 ml of chromotropic acid, which is a solution of 5% aqueous solution of the sodium salt of chromotropic acid, and concentrated sulfuric acid, they are added slowly with the inner walls. Appearance of violet color will indicate the presence of methanol. Let us now consider another test for methanol, which is the Schiff's reagent test. And how this is done? About 4.5 ml of sample is taken in a test tube and 0.5 ml of ethanol is added, so that the sample is fortified accordingly, so that 5 ml volume should contain only 0.5 ml ethanol. 2 ml of 3% KMnO4 solution and 0.2 ml of phosphoric acid is then added. The whole content is then left for 10 minutes. After 10 minutes, 1 ml of 10% oxalic acid is added, followed by the addition of 1 ml of conc sulfuric acid. The contents are then cooled at room temperature, because when you are adding acid it will become hot, and after cooling 5 ml of Schiff's reagent is added and kept for half an hour to observe the color. If there is formation or appearance of purple color, it will indicate the positive test for methanol. Then we have the test for furfural. How it is done? About 5 ml of appropriate amount of sample is taken in a test tube, and about 1 ml aniline and 0.5 ml HCl is added to it and kept for 15 minutes. If there is formation of red color, it will indicate the presence of furfural. There is another alternative method for this. You take about 2 ml or appropriate amount of the sample in a test tube and add 0.2 ml of aniline and 0.4 ml of glacial acetic acid. If the furfural is present in the sample, red color will develop in a few seconds and reaches its maximum intensity in 5 to 10 minutes. So, this is how you can detect the furfural. So, we have studied how these different tests can be done where these compounds that we have mentioned are present as adulterants, whether they are in edible oil or in the gold ornaments or in the petroleum products. Let us now summarize what we have learnt in this module. We studied about oils and fats, which may be divided into various classes based on their origin, vegetable oil or animal fat. The words oil and fat are synonymous, but they differ in their physical state. Let me reinforce here. Oil is liquid and fat is solid at room temperature, but it is due to their different chemical structure. The examination of oils and fats is based on their composition. So, oils and fats are triglycerides of fatty acids, which can be of saturated as well as unsaturated acids in varying proportions. The criminal often use adulteration in oils as well as the gold ornaments as well as the petroleum products. And in alcohol also, methanol is a cheap substitute for ethanol. They will be adding it in liquor and putting it as illegal liquor manufacturing unit. And methanol, we know it is highly poisonous and it can lead to blindness and death. And as forensic scientists, you should know, once you have found these items on the crime scene or otherwise, then how you should be able to analyze and handle and identify whether they were adulterated or not, or what sample and so on. And all these will be giving clues about how to solve this case. So, we studied some chemical compounds, how they are used as adulterants in whether they are edible oils, they can be colorants, they can be different Argemone oil, the lower quality oils and so on. Similarly, in gold, how the copper and the other metals are used. And then we also studied that how we can use the chemical test to identify whether it contains the adulteration or not.